So reviewing the processes of protein synthesis. First of all, here's our DNA double helix once again. It's a double helix, so that just means it has 2 strands and it's wound into a helix form. That was discovered quite a long time ago, that it was that shape. And it's held together by which kinds of bonds hold together those 2 DNA strands? So you can answer that question in the chat if you like. What is the name of the bonds that hold together the 2 DNA strands? Good. Hydrogen bonds indeed. And that's a lucky thing because DNA, we're next getting into replication. Our cells divide with great frequency, some of them anyway. And so it should be easy to break those bonds. And indeed, it is easy to break hydrogen bonds. So one strand of the DNA then is used as a template to make messenger RNA. Pre-messenger RNA in this case, it does go through some processing. Part of that is splicing. And then we did add on in the video the guanine, whoopsie, hang on. The guanine cap and an adenine tail, also known as poly A. So the functions of those, the guanine cap, are a guide to the tRNA and the ribosomes, so that the messenger RNA, which has to meet up with the transfer RNA holding the amino acid and the small and large subunits of the ribosomes. The adenine tail, however, it's thought that the reason that is added, so it's this long tail of just adenine, and it's thought that that is to prevent erosion of your actual messenger RNA before it gets to the ribosome, because there are enzymes in the cytoplasm that could erode the end of that strand. So the messenger RNA is read in three triplets. These are nucleotides: adenine, uracil, and guanine. So remember, for messenger RNA, there is no thymine. Thymine is replaced by uracil. And AUG just happens to be the start codon that signals the ribosomes to start. And UGA is a stop codon. So along comes a transfer RNA with our amino acids attached to very specific transfer RNAs, which have very specific anticodons, so that they can temporarily bind to the messenger RNA.
Come.
Question? Okay. And peptide bonds which are assisted to form by the ribosomes, bind those amino acids together, and we get a peptide. So a peptide, if you can recall back to proteins, we talked about macromolecules. There are dipeptides, oligopeptides, polypeptides. So those are just strings of amino acids. Once you have the complement of amino acids you need to fold into the shape of a protein, then we call it a protein, which are usually very long strings of amino acids. So that was a bit of review. Where does this all take place? So there are ribosomes in the cytoplasm. Some ribosomes are attached to the rough endoplasmic reticulum, and some are free. So maybe I'll just add this here now. So in the cytoplasm, ribosomes are free and some attached to the rough endoplasmic reticulum. So proteins that will be exported from the cell are formed on the rough endoplasmic reticulum. So the RER forms proteins for export from the cell. Generally, that is the case. Free ribosomes form proteins for use inside the cell. So we found before that there are, I'll leave that for a second. We found before that there are attachments to proteins. We've got different moieties. So a protein can be attached to a carbohydrate molecule, for example. They are modified. Proteins are modified by both the endoplasmic reticulum, but in particular, which organelle starts with a G. You can write that in the chat. Which organelle takes that protein in vesicles to the Golgi, exactly, yeah. So, well, proteins have chaperones, interestingly. They are other proteins, so chaperone proteins. They prevent the protein from starting to fold into their functional shape before they're ready. All the amino acids have different properties. So depending on the sequence, they will fold in a certain way. But you have to wait till they're all there. You can't have pre-folding because then the other ones will not have anywhere to go. So they prevent the premature folding of a molecule, and they assist in the proper folding of a new protein. And sometimes they escort the protein to a destination in the cell. There are other proteins that are stress or heat shock proteins, they're called. They're produced in response to heat or stress. So occasionally proteins will denature or lose their shape due to changes in pH or mechanical changes or other chemicals. So they help the protein fold back into their correct functional shapes. Now the shapes of proteins are of tantamount importance to their function. So protein shape, very, very important. So perhaps I'll write a little note here about that. So you can continue writing. Amino acids all have particular properties, such as polarity. Therefore, their order results in specific folding into a functional protein shape.
Maria, I don't mean to interrupt, but
No problem.
Navia's late to class and she just messaged me asking if you can let her in.
Oh, yes.
Oh, sorry.
I think, no, no worries. Let's see. Oh, there she is. Yes. All right. No problem at all. Okay, thanks for letting me know. Yeah, I don't have that notification.
Yeah, no worries.
Okay, so hence chaperones to help that protein fold into the structure it was meant to be. So this slide is good, and this is the slide I want you to take note of some things that I'd like to relate to when we talk about the coronavirus in the cell. For now, let's just look at some terminology. So this structure here, that is your rough endoplasmic reticulum. So it's an endomembrane, but these little purple dots, they are all ribosomes. And let's see if I can make this a bit larger. The protein is starting to form in here. Those are strings of amino acids. They're starting to form the strings, and they're starting to fold as well. Then the vesicle buds off of the rough endoplasmic reticulum, and it's coated by something known as clathrin. Clathrin is a type of protein that coats vesicles. That's its function in the cell. I'm not sure why they require coating, but perhaps so that they don't dissolve. Then the vesicle, this is the direction it's going here, the vesicle fuses with the Golgi apparatus, or the Golgi complex as it's called here. Then within the Golgi complex, it does go through some modification. The modification may be the addition of a moiety, or it may be refolding. And then it buds off there. This vesicle may hold proteins that require circulation in the body, such as hormones, for example, or neurotransmitters. So this vesicle buds then off from the Golgi apparatus and fuses with the membrane of the cell, and the protein is secreted out into the interstitial fluid, which is then taken up by blood and circulated to the body. So nothing ever goes from cells like directly into the bloodstream. It always goes through the interstitial fluid. There's just a small space of fluid between cells and blood. Another vesicle shown here, which is budded off, well, this one has proteins that must be separate from the cytoplasm because they're enzymes. They're enzymes within a lysosome, and their destination will be to digest things within the cell. But they can't be willy-nilly in the cell without being protected by the membrane, because then they'll just digest things randomly. So this lysosome will eventually fuse with a vesicle containing some food. So the lysosome will fuse with a food vesicle, and enzymes within will hydrolyze or digest the food. Hydrolyze. So that's the end of our protein synthesis lecture. So I'm going to stop it there, and then we can look at something else.